Good afternoon to all our viewers, to anybody that is interested in learning more and knowing more about the setting of a life sciences test or examination paper. Today we are going to deviate a little bit from the normal training, from the normal lessons sharing with learners, from the normal memo discussions that we have with teachers over the years so far. As I already said, this afternoon we are going to look a little bit more. You can see it as a crash course in setting papers because we all know that during this time of COVID that schools have to set own papers because of the fact that each school is at a different level of progression with the work and with the depth of teaching. So therefore we thought it well this afternoon to share a little bit with you on setting and then specifically setting the final papers for the end of 2021. My name, Ferdy Venter, and in the Free State I am involved in the subject life sciences. Let us immediately go down to the documents that I want to share with you. And what you will see this afternoon, I'm going to break it down into 3 sections. The broadcast is going to run continuously, but I'm going to break it down into 3 sections. First of all, we are going to look at a very short recap of the guidelines in setting school-based assessment tasks in the FET phase, grade 10 to 12, with the emphasis this afternoon on grade 10 and 11. This guideline teachers in the Free State have received when we did the SBA training a year or what ago. Today we're going to combine that, recap that, but also combine it with the use of the computer in setting these papers. In other words, the first section, a quick recap of the guidelines that you need to use and things that you need to concentrate when setting papers. Secondly, I'm going to share with you the so-called templates or macros that we use when setting life sciences papers to work inside those templates. Many people do not prefer working in templates, but I think after this afternoon's, at the end of this afternoon's broadcast, you will realize how easy your life can become when you use such templates. And then thirdly, I'm going to spend a few minutes, a while, on showing you how to work inside these templates as well as applying some graphics from the computer. Things like drawing tables, drawing graphs, drawing diagrams, editing diagrams in the time that we have available. As I said, a crash course because we are limited in time. Now, when I scroll down these guidelines, and I've adapted it to 2021, our previous training was in previous years, so I've adapted it to 2021, also to the situation where we are currently. Now, before I go there, remember, schools received, and the department of all the provinces, the departments received a circular last week in the middle of October that describes the end of year criteria for writing final papers in grade 10 and grade 11. We received the circular from National. It was the latest one was circular S17 that retracted circular S16 and circular S17. I'm not going to show that today, this afternoon to you. You have it at your schools. You have it in your offices. That one also refers to circular S7 of 2020. In other words, all the guidelines needed for the end of 2021 will be found in circular S7 of 2020. But as we progress, we are going to tackle many of those criteria set by the DBE, so you will be very comfortable when after today you have to start setting. If you have already started setting, if you have already completed your papers for the end of the year, it's a good thing that you have a look this afternoon and see if you are in line with the guidelines. I'm not going to spend too much time on introductions and reading through documents. I want to move to this part that says steps in setting question papers before you start. Now, it might sound to some people, why do we start here? I know where to start, I know where to look at and what to look for, but let us cater for all people this afternoon that wants to start to set a paper from scratch. I mean, it is a YouTube video that is rolling, so if there's things that you know already and things that you are comfortable with, you can just forward to a next part of our conversation. First of all, very important, you should know exactly which content should be covered and ensure that learners know what content they must prepare for that particular SBA task, in this case, final assessment tasks for the end of 2021. You must keep in mind, first of all, the taxonomy. That refers to the degree of difficulty of each question and is dealt with in the section on taxonomy that will follow later. Everything must be checked and controlled by a moderator. At schools, you have your internal moderators, your departmental heads. I always say you can very easily swap with a neighboring school, you and the teacher there, if there is not a moderator at your school or a life sciences departmental head at your school, swap with a neighboring school, moderate one's papers in a so-called pre-moderation, but also afterwards share some of the learner scripts, mark some of those learner scripts, swap it and return a small report or a short report and sharing some of the concerns. And from there on, you finish your marking. It might take you an hour or two doing these things, but I promise you, when these things are in place, when moderation is in place, your standard at your school will be of a high level, and also the papers will be accepted by provincial officers so that your learner's progression is then valid and reliable. Examination instructions should be put at the beginning of the question paper so that learners can see which instructions they must follow. You must formulate your questions and instructions very clearly and keep the language simple. This is a very important bullet. Keep the language simple. For all our learners, many of us struggling with English, our papers are in English currently, and in Afrikaans, so therefore keep the language simple for your home language speakers as well as your first additional and second additional English language speakers. Don't try to make your paper difficult, or you think you will raise the standard by making use of a complex type of a language, difficult type of English. You are not raising the standard of your paper doing it like that. Keep the layout of your paper consistent and according to the formatting instructions. We will come to that. Be sensitive to language and spelling errors in your paper when you set. Ensure that your questions might not be interpreted wrongly by avoiding ambiguity. Prevent yourself from repeating questions from previous papers. Yes, we tend to do that. We tend to get an idea from a previous paper. That's no problem. You can even use diagrams from a previous paper. But then please change the labels, put in some new labels, and also change the questions below that table or diagram or graph, so that you don't just repeat your questions, because our learners, they find papers all over the place. They do that for revision, and what is then the challenge if they come across a paper that they can answer straight from knowledge that they already obtained. Do not repeat questions in the same paper. Careful for this one. Sometimes in your section A, multiple choice, you ask something, and then in section B, you ask another thing that is more or less the same, or you test the same knowledge component, or you are testing maybe the same skills over and over and over again, be careful for that. Of course, we will later on see that you indicate sub-questions marks very clearly. Should be a correlation between the number of facts required and the mark allocation, we know that. And then that long paragraph that explains that in case of a question having more possibilities, then the mark allocation, only the number of answers relevant to the mark allocation will be credited. So if you ask, for instance, how is the leaf adapted for photosynthesis in grade 11, and they must just describe, let's say, 3 adaptations, and then you make it count 6 because there's more, then learners must know only the first 3 will be marked. If a question stipulates, give 2 reasons, explain 1 adaptation, then only the first 1 or the first 2, whatever is asked, will be credited. So learners also mustn't waste time in answering 2, 3, 4, 5 things when only 1 or 2 has been asked. Now, questions must be user-friendly. User-friendly doesn't mean it's simple, it's easy, but user-friendly, as we already said. Make the language understandable, and then you will also have user-friendly questions. Stay away from biasness. Candidates with different backgrounds need to be catered for example, learners with different terms of reference, language, religion, disabilities, gender, race, etc. Keep it neutral. Keep your papers neutral. Or if you want to go to a particular reference, then have a balance between all the references in our country. Choose your verbs carefully. We will come to action verbs. Each question has an action verb. Is the question state or describe or explain or compare? Choose that carefully, because when they have to state, it's nothing else but mention or name. Describe is a description, as it says. Explain, we will come to that, but explain, if I say explain 1 reason, then I give the reason and I explain it for 2 marks. Explain 2 adaptations. Then you mention 2 adaptations and you explain each of them for 4 marks. Compare, remember, compare can be similarities and differences. So you must decide, am I going to ask comparison in a table? We will come to tables. Or will I ask them to describe differences or mention similarities? Be specific in your question. Important, learners must be familiarized with examination vocabulary and terminology during the course of the year, of course. Of course, if the learners are exposed to these things throughout the year, you can assess them on that at the end of the year. You can't at the end of the year assess things that you have never been referred to or mentioned or taught throughout the year. Now, the methodology in the setting of a paper. And I've just highlighted a few things, but we can go through all of them. Follow the suggested sequence in setting questions. We will come to the format of papers a bit later on, and then you will see what I'm referring to. Determine the topic of a particular question. Now, for that, you need a grid. You need a taxonomy grid. And your grid, you will later on see, will be divided into content, into cognitive levels, and into difficulty levels. But if you decide on a topic for a particular question, what I mean with that is, let's say section B, question 2.1 is about ecology in grade 10, then that topic then the questions is about that topic. If your question doesn't refer to the diagram or the table, it must be moved to another question or a question on its own. Refer to the particular section in caps in your work schedule or your examination guidelines. Grade 12, we are not discussing grade 12 this afternoon. The other 2 grades will receive very soon examination guidelines for them. But in the meantime, remember, work schedule, we nowadays actually use the ATP, the Annual Teaching Plan. And the Annual Teaching Plans, grade 10 and 11, has everything. It's a work schedule. We have even the caps explained in there, the content explained in there, as well as the assessment plan, to tell learners the formal assessment plan. So you refer to your ATP. That must be open next to you or on your computer when you work. Because if I can give you the sequence right now, there on top it says follow the suggested sequence. You start with your ATP, you start with your CAPS in grade 10 and 11. What must learners know on biodiversity in plants, for instance, in grade 11? If I know what they must know, then I can work on a question. I can't just go to a textbook and pick a question or a homework piece and put that into my question paper, but that is not even in CAPS. It is not even expected for learners to know that. Fourth bullet, as you proceed, ensure that you remain within the framework of marks, question types and sections, applicable content, and cognitive level, as well as nowadays in the amended CAPS, difficulty levels. Fit every question into your taxonomy. In other words, every question will cover certain content, but it will also cover certain difficulty levels and cognitive levels. I will show you later on. So please stay with us. I will be to the point and I won't waste time today, but because I know these things also take data to look at it. But make a plan and stay with us. You are going to find lots of very maybe interesting and informative information later on. Try to fit the question into a relevant real-life situation. You see many of these questions start with a certain animal living there or a certain plant growing somewhere is doing this or showing that, and then questions on it. Especially in the grade 12 final exams, you see many of that. So even in 10 and 11, make it interesting to the learners that they also enjoy answering these question papers. Think in a way that the teenage candidate would think. Make it as authentic to them as possible. It is advisable to generate the memorandum and cognitive or content grid together with the setting of the questions. I cannot emphasize this enough. While you're busy setting your paper, if you work on the computer, split your screens, open two documents, your paper and your memo, or marking guideline then for that matter. And when you identify a question and when you create a question, formulate a question, immediately write the memorandum. Because if you ask, describe the adaptations of a leaf for transpiration, go and you say, okay, this is 10 marks. Sit down, write the memo or the marking guideline, compile that, and see if you can get to 10 marks. If you can't, the learners will definitely also not. So then if it's only 4 marks that you think is good, or 3 marks, or 5, then you know what your maximum subtotal for that question should be. And then the cognitive grid also, you will see later on. When you keep that up to date, you will immediately see where am I beyond certain cognitive levels or behind maybe. So that when you need a certain type of question, you also know in which cognitive category you must classify that. Now the detail and the sequence of procedures will be dealt with in sections that's coming. But this is more part of the detailed SBA training of the past. Today I'm just going to quickly refer to these things. So yes, these are some background info that you need before you sit down setting your paper. Before I go to that, I started here by saying follow the suggested sequence. Now, a good thing is to sit down, get yourself a rough piece of paper, and just draw a rough program or a skeleton for yourself on your paper. In other words, what I normally do, I take a clean A4 sheet of paper, and I write quickly the section A, multiple choice, biological terms, column A, column B, matching columns, and then I write section B, question 2.1, 2.2, 2.3, etc. I leave myself gaps in between, and then do the same with question 3. And remember, there's no essay anymore. So we at the end of question 3, 3.1, 2, 3, 4, 5, wherever, is the end of your paper. And then I open the content that I need to cover. And I very roughly divide that content into the skeleton of mine. 2.1, if it's grade 11, I will go for photosynthesis questions. Let's say 8 marks and sub-questions on photosynthesis with a diagram. And I write down more or less what I have in mind there. My second 2.2 will be on respiration, grade 11. And I'm going to ask them a investigation maybe on carbon dioxide release in cellular respiration, with my subtopics and more or less marks. So, and that I do with the rest of my paper, with all the sections. And then, and listen what I'm saying. I'm starting with section B. Because when I slot in these rough questions into section B, then I can come back and just fill up gaps in my content and cognitive demand by then putting in my multiple choice. I leave my biological terms for totally the last part. Because when you have, let's say, 10 biological terms, then you have 10 one-mark things that you can slot in where you need content or where you need cognitive levels. All right, that is a set cognitive level. We have to think about that, and I will tell you that later also. Right. So then I have my skeleton, and from my skeleton, now I go to the computer. Now I go to my macro or my grid that I have, or my template, and I start slotting in these questions, typing them in, or getting graphs or diagrams from wherever, and then build up the questions. Now, viewers, you cannot set a paper when you don't know what to set on. Now, earlier this year, we gave teachers in the Free State, after a discussion with people at DBE, sorry for this, we have this program of formal assessment for this year. And I'm not going to refer to all this in this table. But grade 10 and 11 has exactly the same distribution up to the end of the year, and then very clearly now also. Unfortunately, teachers and learners know this right from January 2021. And that is explained also in Circular S17. The distribution of year mark, in other words, SBA mark, and final assessment mark, is again this year 60-40, 60% versus 40%. In other words, of the 100 marks, the percentage for promotion, 60% of that is your school-based assessment, in other words, your practicals, your tests throughout the year, the assignment, etc. And then the last 40% is on the final assessment. Now, in the past, before COVID, it was 25-75. You will all remember that clearly. 75 exam, 25 SBA. But because of the challenges and the restraints of the pandemic, we, for the last 2 years now, moved to this distribution. Now you see something very interesting here in red. After a discussion, also with our colleagues countrywide in our National Subject Committee, we have decided and we have agreed, also with our colleagues at National, that we cannot stand in the way of a school or schools that completed the full curriculum and that wants to write a fully fledged paper 1 and paper 2 with grade 10 as well as grade 11. That will be allowed, with, of course, the recommendation in the circulars that we go for risk-adjusted formats, which means 2 tests. So schools must think very, very carefully when they sit down and make this decision. Think of the workload of your children going to study for 2 fully fledged papers, workload writing 1 test of all the topics of the year. So there's challenges in both. If you take this route as a school, then your papers can be 150 marks for each paper, and your duration will be 2.5 hours for each paper. And before I forget, in the Free State, you will receive a common timetable from the exam section. all the subjects will be included in that common timetable. That doesn't mean common papers. It only means common dates. It means, and you will see for life sciences, hopefully when the timetable in its final stage is the one as we've discussed it in the draft form, life sciences will have 2 dates. Then if you are writing, and the dates will indicate one date for grade 10 and one date for grade 11. And that means when you go this route, then you can write your grade 10 test on one day with the learners and your grade 11 test on the second day. If you go this route, full papers, then the school will adapt itself so that maybe then on the first date, if you use the common dates, of course, on the first date, you will then write maybe paper 1 of both grade 10 and 11, and on the second date, paper 2 of grade 10 and 11. But that is up to the decision of the school management team and the schools themselves. So I am going to repeat it. Common dates will be provided, but not common papers. Schools must set their own papers. Now, the risk-adjusted format for schools that did not complete the curriculum. Complete doesn't mean you didn't get to the last 3 weeks of the year. Complete means throughout the year. There were topics that you maybe just touch on, and then you have to move because you didn't see the learners for a week or 2. And then when they come back after 2 weeks, you carry on with the next topic because you were afraid you're going to fall behind. That means you did not complete the curriculum. Completed curriculum means all the content in the ATPs, plus the informal activities, the homework, the classwork, the class tests was done and completed. So schools that did not complete, they will have big difficulty in writing 2 fully fledged papers. So this is where the risk-adjusted format comes in. As the circular indicates to us, grade 10 is expected to write for 1 hour, and in life sciences we know that 1 mark means 1 minute of writing. So if they write 1 hour according to the circular, then that test should be 60 marks. But you will see in a few seconds how we distribute that 60 marks. Grade 11 will write a 2-hour test on 120 marks. Right. And we really plead with schools and teachers not to deviate too much from these 2 options, A and B. You are also going to receive in the coming days, you are going to receive what we call assessment frames or assessment frameworks. I know we have provided you with a draft just to show you where the thinking is going. But you must get rid of that draft because there's much more new information in this document that I'm sharing with you now than that was in that one. And don't be afraid, we will, after this broadcast today, I am going to distribute this same document, this one in front of you, to the schools, to the subject advisors, and through them to the schools. So you will receive this in the next day or 2. And then, of course, the official document will come with all the subjects frames in it. This is only for our subject, life sciences. So don't deviate too much from this. If this is given, I think you must stick to this guidelines and criteria. Now, grade 12, we're not going to refer to this afternoon. Now, this further elaboration I took straight from the 2020 guidelines on promotion requirements for life sciences. I copied it and I pasted it into this document because this was what was given from national side end of last year, and it is also applicable to this year. For promotion requirements, I must very easily make it a /2021 because that is what it is. When designing the tests, the life sciences teachers must ensure that, okay, we have said this, I'm not going to repeat things. The test should cover all curriculum content that was taught, including content covered in term 1. Content not taught cannot be assessed. We really understand that. But be careful not to let your children only going to write on term 4 work or term 3 and 4 work. Because in 10 it's easy, because term 1 and 2 is paper 1 and term 3 and 4 is paper 2 for a full-bodied paper. But for one test paper it's not so easy. So assess them on the work of the year. And again, you will see in a minute or two, we have given you, we gave you some good hints, some good suggestions on what to ask in a particular topic. And I will explain when we come there. Use your professional judgment to identify your core or fundamental content. We will help you with that. The tests are balanced in terms of cognitive levels, degrees of difficulty and content. We spoke about that. Weighting grids are used. We spoke about that. It follows the NSC, National Senior Certificate. That is the end of grade 12 examination. It follows those exams, design and rigor. In other words, the format, the layout of your paper, and the rigor, the feeling, the heart of this paper should be in the same, along the same lines than a final paper in grade 12. Each test paper must cover all the specific aims. There are 3 of them, with specific aim 2, 20% of the total paper. Try to adhere to this criterion. It is the scientific investigation part. Your experiments and questions on that, your aim, conclusion, validity, reliability, those type of questions. The test must be administered under controlled conditions, no question about that, and it must adhere to protocols of pre- and post-moderation. It's very, very important. I spoke about it earlier, I referred to it. Your paper should be pre-moderated. A tool should be completed to say this paper is along the lines, is adhering to all the criteria, and then post-moderation on the learner scripts. No provincial or district common tests are administered in grade 10 and 11. Now, the components of a life sciences paper. There's just a few ideas, and I'm not going to spend a lot of time on this. You can refer to that when you start setting, and I'm not going to explain all of them, but these are the type of questions that you can make use of. Graphs, visual questions like diagrams with labels, a picture maybe from an article from a magazine, correct terminology, multiple choice, problem solving, etc. I removed the part that says essay, because if this was for all SBA tasks, maybe in future we will definitely use that essay in the source-based essay in the assignments. But we are done with assignments this year, so an essay is not applicable to these papers. Then instructional verbs. How to start your question. And I'm not going to explain it. We did it in the training. There are a few verbs that you can use. Now, again, keep it learner-friendly. Keep it user-friendly. They are used to calculate, maybe compare, definitely define, describe, determine. Not so familiar with differentiate. They are familiar with discuss and explain and identify and label, as well as list and mention. Name, state, suggest, tabulate. Yes, they know these things, but be careful for things like analyze, classify, contrast. And you can look at the explanation for these verbs. Remember a tabulation. I will explain that again when we come to drawing tables. Tabulation, if you say compare or give the difference between two aspects, tabulate the difference between two aspects, then the table, the learners get 1 mark just for drawing the table, and then the other 4. For each difference, 1 mark, it's 1, 1, it's 2, plus another 1, 1, it's another 2, so they can get then For comparing two things, they can get 5 marks for that table. When they compare one thing, it's 3 marks for that particular table. So I'm not going to spend time on the action verbs. Now, the things that you are waiting for this afternoon, I know that. The format for papers for the end of this year. Schools that completed the curriculum, as we already said, and want to go the full 2 papers, grade 10 and 11, here's the format. Again, section A, not negotiable. You cannot ask them 60 in grade 10 biological terms for 60 marks. That is not the rigor and the format and the feeling of our final papers. A section A of 50 marks. And then 2 section B questions, 2 times 50 for 150 per paper. And then the duration of that paper will then be 2.5 hours. Now, you know these things, multiple choice questions, biological terms, column A, column B, pairing off, or no, not pairing off, it's not pairing off, it's that statement given and then a choice between 2 answers. And then section B, 2 questions, question 2 and question 3, each divided into 2 to 4 subsections, as we have it. You know these things. Now, the schools that go for the risk-adjusted format, schools that did not complete the curriculum, of course, you will ask, I have completed my curriculum, but I opt and I choose and my school feels it's better to write a test as given by the circular. Please do so. You don't have to go to 2 papers if you completed the curriculum. You can still write only the 1 test in 10 and the 1 test in 11. But then please keep to these criteria. 1 hour for grade 10, 2 hours for grade 11. And I've split it so that you can understand it very easily. Look at this table. Same distribution, but then grade 10 for that 60 marks will have 3 20-mark questions. Section A 20 and section B 2 times 20. Now each of those 20 in section B, of course, these 2, each of them will be divided into subsections. 2.1, 2.2, and 2.1, of course, let's say 2.1 is a graph. And then you have 2.1.1, 2.1.2, 0.3, 0.4 on that graph. That's a subsection that we refer to. 2.2 will be the second subsection. And then the same with grade 11. For 120 marks, it's 3 times 40 marks questions. 40 for section A, 2 times 40 for section B. Same story. That 40 in section A, of course, comes from these different types. And go to past papers. If you're a new teacher or you're there alone in your school and you don't know who to ask, and you are not phoning us, one of the subject advisors or myself, feel free. But then you go to past papers. You go to any past paper in any grade and you will find exactly the same format there. Of course, the mark allocation will be, in a test it will be different, and in an exam it will be different. But then you have the idea of the 3 question types. Now, very importantly, allocation of content, and that is for November 2021, ladies and gentlemen. We don't know what's going to happen next year, how it's going to look. Are we going to run normal timetables, no rotational systems? Then it's a total different story. This is it's for now. It's for the coming November examinations. In the Free State, we have decided, we have sat together, we had a meeting, and after a good discussion, we had a consensus on the following. I'm just going to make it a little bit smaller so that you can see the big picture. I hope everybody can still see there. I will enlarge it again. For grade 10, for the risk-adjusted format, These are all the topics in caps, for that matter in the amended caps, for grade 10 for the whole year. And as I said, fortunately grade 10 is divided into paper 1, there you see them, the P1s. In the amended caps, these are all the paper 1 topics. And then paper 2 topics. Now, as I said, fully fleshed papers, then paper 1 will stop there, and paper 2 will start there and stop there. I hope my cursor is on the screen, but if not, then paper 1 is all the paper 1 topics and paper 2 is all the paper 2 topics. And here are the original marks in the amended caps. In other words, you can go and check. We can make errors, of course, but we have really tried to get it exactly as in caps. Chemistry of life, 33 marks out of the... Why do I say 300? Because it's now 2 papers combined in 300 marks. Yes, if you're going to write 2 papers, you will split it up, 150, 150. But just to show you where we come, where we got the marks for the test, we had to show you this. So these marks were converted for each topic for the risk-adjusted test of 60 marks. So chemistry of life, and we try to stay away from 2 marks and 3 marks. So we converted, but here and there maybe we add a mark or 2 as to make it a substantial amount of marks. Chemistry of life, 8 marks. Cells, 10. Cell division, mitosis, 10 marks. Plant tissues, 6 marks, etc. I'm not going to read the whole lot of them. But look what we've done. We are recommending that you can leave support and transport in plants. In other words, the transpiration things. Why? Because these things in brackets, the red ones, this is basic knowledge. This is prerequisite knowledge for the next grade or grades. Grade 10 need these red things in 11 and in 12. If you do it in grade 10, they need it in 11 and 12. If you did not get to some of these, you will have to adapt your paper accordingly. But at least try to cover these things in brackets. So if you must ask questions on chemistry of life and you doubt, now, must I go inorganic? Must I go vitamins? Must I go fats, lipids, carbohydrates, and proteins? We guide you by saying, ask questions on organic substances. In other words, lipids, carbohydrates, and proteins. Why? Because in grade 12, they're going to need this information when it comes to the building up of nucleotides in DNA and RNA and those type of questions. For 8 marks. When you do the cell, when you ask questions on the cell, and before I forget, please, schools and teachers, give your learners a scope of work to study, to prepare for this coming test. Don't hide from them and tell them you go and study all the topics in your textbook. They will never know where the emphasis is. So compile at your schools, according to your paper, compile a scope of work for your learners. Don't give them the questions or the answers. I don't even have to say that. But give them a scope of work. So in other words, you tell them in chemistry of life, you go and study organic substances, whatever that entails in grade 10. Basic units of life. Then you tell them, focus on the nucleus, the mitochondrium, and the chloroplast. Because the chloroplast they will need in grade 11 in photosynthesis. And the mitochondrium and nucleus they will need, and the chloroplast in grade 12, again, for where DNA is located. And when it comes to, in the nucleus, the structure and the whole story of protein synthesis in grade 12, translation and transcription. They will need basic knowledge on these 3 organelles, at least. And if it's 10 marks, I mean, now it becomes easy. I must cover 10 marks with those 3 organelles. Cell division It's the basis for meiosis. So here it's 10 marks. You must ask questions on location and phases of mitosis that helps your learners to prepare for grade 12 meiosis. Plant tissues, transport and chlorophyll containing tissues. In other words, your xylem, phloem and all the parts containing chlorophyll. Concentrate on them. And in animal tissues, concentrate on the nervous tissue, because we have a topic in grade 12, the nervous system, where they need to know the basic structure of a nerve cell. Plant organs. Concentrate on leaf structure and then the cells, the cell functions related to photosynthesis, because they need it in grade 11. It's only 4 marks. You can play around with these marks, viewers. You can, if you maybe want to go 6 marks here and 5 there, that's not the end of the world. This is a basic suggestion or a skeleton for you to compile your papers. Then we thought support and transport in plants, you can leave that out if you want to leave out something in grade 10. Support system in animals, this human skeleton, you can leave that out in grade 10 if you want to. But if you want to ask questions on it, by all means, you're more than welcome. Transport systems in mammals. Remember, it's all about the heart, the parts of the heart, the types of blood vessels, the structure of blood vessels, the function of different types of blood vessels and places in the body. Now think carefully, the heart itself, yes, it's nice. You have taught it in class, but you need to assess it now, because the heart itself is not going to be covered in any other grade to come. But the types of blood vessels in thermoregulation in matric, for sure, they need to know the difference between a vein, an artery and a capillary, and how it looks, the structure of it, and the suitability to do its function. Ecosystems, it's a difficult one, because where in grade 11 and 12 do we do again ecosystems? But nowadays, in the current environment that we are living in, we thought it necessary that learners should at least be assessed on different biomes and maybe the difference between the abiotic and biotic factors. And if you want to go and have enough marks to cover, maybe something on food chains or food webs. And remember, this is a food chain or a food web can be a very nice question in multiple choice, where you give a food web and then you ask just a few multiple choice questions on it, or one multiple choice question on it. So it's 2 marks. You spend 2 marks on that food chains and webs, and then the other marks can go to other questions. Biodiversity and classification, it's only 5 marks. Main groupings of organisms, because they need the groupings, they need classification systems in grade 11 in biodiversity of plants and animals, and also in evolution in matric, when it's about the classification of the hominids. So it's good if you can assess it. And then history of life on Earth. Some people will say learners need that info for evolution in matric. Go and have a look again and think twice. We think maybe in this assessment you can go without it, because it's at the end of the grade 10 work. You have to catch up on all these others maybe first before you can get there. So it's maybe a good topic just to not to ask questions on. So that's the grade 10 framework. Let me make it a little bit bigger. There you have it. Going down to grade 11, let me make it a little bit smaller so that we can see all of them. Grade 11, because it's a more senior grade, and because it's 120 marks, and because we know that many schools manage to complete the curriculum in grade 11, but not in 10, we thought, okay, let's leave all the topics, let's assess all the topics in grade 11. And you will see more or less balanced, 12, 12, 10, 12, 15. So it's a very balanced distribution of the topics in 11, even if this marks do not 100% convert to those marks. So again, topics put in brackets in red are those that we think will be necessary for these learners to be assessed on for grade 12 next year. So in photosynthesis, in photosynthesis, you concentrate on questions on things like the importance of the process and then the influence of different factors. And then, of course, here you have an investigation. Is light, I think the investigation that we recommend is light necessary for photosynthesis, if I'm not mistaken. And then also in respiration, later on, there's one investigation. The gas that is released, carbon dioxide, is released during respiration. So those are your two main topics for your scientific question in grade 11. Your question that goes to variables and aim and conclusion and those type of things. Then when you go to animal nutrition, you concentrate on glucose control. Why? Yes, it's fine. Ask them the alimentary system, ask them functions of the liver and the stomach and whatever. But go at least to glucose control, because in grade 12, that is the basic information they need in homeostasis in grade 12 of the control of glucose. So you have to cover, and then in your scope, you tell them that. You tell them, there's going to be questions on the human alimentary system, but then there will also be questions on glucose control, so that they go and study that. It makes your work easier next year in grade 12. Respiration, difference between aerobic and anaerobic respiration, plus that one investigation I referred to. Gaseous exchange, the important one here is carbon dioxide control. Carbon dioxide control. Again, homeostasis in grade 12. Excretion in humans. Yes, ask them on the kidney and the nephron and filtration and reabsorption and all those, if you want to. Then just give it in the scope for the learners, but then also make sure that you ask something on water and salt control. I mean, again, if I must think quickly, in section A, if those two, maybe glucose was in section B, like a longer type of question, a 4 or a 5 mark question. So you can't go to another 4 or 5 mark water and salt control. Then you ask in section A, for instance, the hormones responsible, or the target organ, or the gland that secretes, because they must know it in grade 11 already, because it's part of the control function. Now, these are all paper 1 topics, but not necessarily the order in which they were done this year, because remember we started the year with classification of microorganisms and then plants and then animals. But here it is divided into the 2 papers, as well as the initial marks in the amended caps. And there's the recommended marks for your test of 120. Now paper 2, Biodiversity and Classification of Microorganisms, we thought it will be good if you assess the role of bacteria in biotechnology. Why? Because of the genetic part, the genetic engineering part in grade 12, where this becomes crucial. Biodiversity of plants. Remember in our ATP, and after visiting some schools, myself and the subject advisors, we found that teachers, either they don't really understand what is meant by this comparative table, or how to put it in a workbook. It's exactly what it is. It's a comparative table, and the plants in the caps, it explains. Learners must know the difference in the plant groups, the bryophytes, the pteridophytes, the angiosperms, the gymnosperms, things that they have to compare, things like transport tissue, xylem and phloem, is it there or not? Seeds or spores, is it there or not? Those type of things we compare between, and if that table is drawn, the learners go and study that table, you can ask a lot of questions on it. But then also maybe the structure of a dicotyledonous flower and the functions of parts in reproduction, because that becomes important in grade 12 when we do meiosis and we do a little bit of life cycles, the change from diploid to haploid, and then again from haploid to diploid. If learners know the parts of the flower, at least, and where does meiosis take place, and where does pollination in this part place, and fertilization, they can distinguish later on in grade 12 also. Animals, biodiversity, comparative table again. Go and look in caps. What has to be compared? What has to be compared? It's the body plans, triploblastic, diploblastic, the symmetry, etc., etc. 12 marks for that. And then phylogenetic trees. We know that phylogenetic trees is important in plants as well. CAPS expect that. But if you then don't ask it there, we gave the option here in animals. Ask a phylogenetic tree, nice 4, 5, 6 mark question, or even a section A question, so that in grade 12, in evolution, they don't have problems to know what exactly does a phylogenetic tree tell us. Then population ecology and human impact, the last two topics that many times in the 4th term in grade 11, people sometimes rush, people struggle to finish, they quickly go over it. Fortunately, nowadays, we don't have human impact in grade 12 anymore, so this is the place where you should do it. You can't just ignore it now totally. So population ecology, we thought factors influencing population size. In other words, it's the parameters, immigration, emigration, and then also, I can't get to the other two, the birth rate and the death rate. My goodness, it will come to me soon. Human impact on the environment, the last one. Mortality, natality and mortality. Those two, the death rate, mortality, and the birth rate, natality. And of course, immigration and emigration, the factors influence population size. And then growth forms. Here's a nice graph that you can ask. The J-curve or the S-curve, and questions from that. And there's 15 marks to play with. Human impact on the environment. Now, this is a difficult one. It's only 15 marks. There's how many factors? There's climatic factors, there's water, there's biodiversity, there's solid waste removal, etc. We suggest, focus on the topics that were taught in your class while using the mind map of human impact. We gave you a mind map years ago, especially for grade 12, but also to use in 11. Focus on the core things in that mind map and ask questions in your exam paper that you did in class. If you did only 2 of them for some reason, carbon dioxide or gases and maybe solid waste, then you ask 2 questions on those 2 topics. So, viewers, that is the content of our papers. About an hour has passed. I think we must take a quick break of 5 minutes. And then we will carry on by keynotes in designing and then also come to a particular paper using a grid and also how to draw some of these things in your papers. We will be back in 5. Welcome back to our presentation this afternoon. For those of you that have maybe joined us a little bit late, we this afternoon in thinking about the final assessment in 2021, we are talking about setting grade 10 and 11 papers. So please go back in the presentation, you can rewind, you can forward on your YouTube video to see what we have said so far on the content, on the way where to begin a paper, on the allocation of marks, on the suggestions given on question papers and how many marks. Now we are looking at the taxonomy. And remember, when you receive this document, I'm going to send this document after today, early tomorrow morning, to the subject advisors for distribution to schools. There will be things that you find there that we didn't do this afternoon, that we didn't look at, because that was part of our training a year or two ago when we did SBA training. So this is like, as I said earlier, a crash course in that training, but also in combination with our setting the end of 2021. So when we set a paper, we need to know about Bloom's taxonomy. And I'm going to the paper immediately so that we can have a look at the content there. And I'm not going to take a lot of time explaining this because it was done properly in our training, in all the districts. But just remember carefully, there's 4 levels. And again, some subjects talk about level 1, 2, 3, 4. We talk A, B, C, D. Some groups or even subjects has 6 levels, others has 3. Doesn't really matter, but we use this grid. We use this rubric for determining our cognitive levels. On level A, and there on the right-hand side, is the mark or the percentage allocation in your paper. It means that if your paper is 60 marks, 40% of that paper should be set on cognitive level A, 25% on B, 20% on C, and 15% on D. And I will show you, I already worked out the grids, the templates where you're going to capture your setting for the different papers, for the 60, the 120, and the 150 mark papers. Knowing science is on level A. People, this doesn't mean A is easy, B is difficult, C is more difficult. That is why there's difficulty levels. A means it's a thinking level, it's a cognitive level. Straight from the textbook, straight from the notes. I know science, I know life sciences. These science can always be replaced by life sciences. I know my work, I can immediately recall straight from a book. These are things like, there you see examples, list, name, give labels, identify, define. Straight from the textbook, straight from the study guideline or whatever. Understanding science is now, I know a certain part, but now I also have to understand it, think deeper. Good question is, give the number or the letter and the name of the label that does the following in a particular part. Now you must think further. I know this label's name is the pollen, but now I must understand where pollen comes from and how it plays a role in reproduction in this plant, and then I must give the function or whatever. So I have to understand a particular topic. I'm on level B. If I apply that knowledge, how will that pollen play a role in cross-pollination versus self-pollination, etc.? Now I'm comparing and applying my scientific knowledge, or in a different situation. We always say it's applying to a new situation maybe. That's not directly in the textbook. Then we're applying, we're on level C. But now you see here things like predict, apply, use knowledge, demonstrate. I will tell you in a bit the cognitive levels of certain typical questions in our question paper. And the last one is on the highest cognitive level. Now you evaluate, you analyze, you compare, you synthesize scientific knowledge. Select, differentiate, analyze, etc. It's on the highest level. That's why it has the less amount of marks also. So that in short, the cognitive levels. The degrees of difficulty, which is also now in our caps. So apart from thinking levels, a thing can be on level A, straightforward from the book, but it is a difficult or a very difficult concept. Then it is on level A, but on difficulty level 4. Something can be on level C, applying knowledge. It's a high thinking level. Let's say if I go even to D and I ask, give the dependent variable in this investigation. They're always on D. Variables. But it's straightforward. From the information given, the learner can see, oh, the dependent variable is the amount of water being released by the leaf. Very easy. But it's on level D. As a thinking level, it can be an easy answer. In other words, level 1 for difficulty levels. So there's your difficulty levels. Easy, moderately challenging, difficult for the average learner to answer, and very difficult for the average learner to answer. So keep that in mind. I will show you in our grid, or our cognitive grid, taxonomy grid. Now, before we come to the computer skills, let me show you such a grid. And what I've done, as I just said, let me show you just this enlargement, and then scrolling down. Okay, let me explain this from top to bottom. We have done it in the training, but again, here's your question numbers, question 111, 112, 113, etc. In other words, on the left-hand side, those questions forms part of section A, multiple choice, always in our papers in that order. Then 121 up to 126 are biological terms, 1.2 of section A. 131, 2 and 3 is the column, is the A and B, or either both A and B, none, A, just A, or only A, only B. And then you have 2 questions or 3 questions, 1.4 and 1.5, or 1.6 sometimes, that has one-word answers. Very carefully, one-word answers, state or name or label or give for then the rest of section A's marks. Now, this amount of marks, this amount of question numbers, you can play with in your questions, ladies and gentlemen. You can play with this. You can only ask 6 multiple choice questions, you can ask 4 bioterms, etc., and then go more on the others. No problem. This is just a guideline, please. Then, when you set a question, your first multiple choice question is a very easy one. Now, listen to what I'm saying, easy. So then I go there, degrees of difficulty, easy. But remember, multiple choice are all 2 marks. So it's on level A, it's straightforward. And normally we start with an easy question. Let learners become comfortable, they read the question, they know the answer, they answer the question. It's on cognitive level A. It was on nutrition, and it was very easy. Now there's my 3 2s, because multiple choice are all 2 marks, and I've graded it accordingly. My next question is a little bit more on a higher level. It's on level B, never mind what it is. It's on photosynthesis, and it was a difficult question for the average learner, and I grade it like that. Can you understand the way that we grade these things? Let me just center those things, so because I see some of them are not in the middle. Right. So, multiple choice can be a variety of cognitive levels, variety of content, and a variety of degrees of difficulty. When you come to bioterms, biological terms, before you even start setting your paper, you can do the following. Because they are all 1 mark, and they are all on level A of the cognitive demands. Finish and clear. Then you decide which topic they are, and the difficulty levels can differ. You can have a very difficult, for the average learner, biological term, or you can have an easy, straightforward one. Those, there they differ. When you go to, is it A or B, only B, AB, whatever, they are all on level B. I can promise you that. You don't even have to think twice. So you add your 3 2s there, even before you start. But here you play around with content, and you play around with difficulty levels. And then 1.4 and 1.5, it will differ. There will be one there, maybe one on C, one on what, depending on what you ask. If you ask, let's say your investigation is here in 1.4, your investigation on is carbon dioxide released through respiration, then you will have, if it's an investigation, mostly all investigation questions, they come there on level D. Variables, aim, conclusion, those type of things. So that's your section A. You grade it according to this. And when you go down, here's the totals. Look there. There's that, let me start here. The norm mark the norm mark is according to our cognitive demands of percentages, 40, 25, 20, 15. The norm mark out of 150 for this, 120 for this test is then already calculated for you 48, 30, 24, 18. And when I slot them in on top, they immediately calculate down here. So when your grid is done of your whole paper, then you should be as close as possible to that 48 on level A, that 30 on level B, that 24 on C, and that 18 on D. The same with those, they immediately calculate themselves. There's the norm as it should be, and there's the marks. Then remember, this is percentage. Those are marks out of 120, and then they total as you slot them in on top. And even the content slots in. So when you've done this, and you see, but I need a lot of questions on gaseous exchange, then you know where to put them and you add them here, but they must be on level B. Then I look for gaseous exchange questions on level B and I put that in my paper. So can you understand why I say keeping this thing up to date with your memo when you set is crucial. So I hope everybody understands the totaling here at the bottom. Don't worry too much. I believe that our calculations are correct. So when you slot in on top, at the end, they should all count up. And you see, here's the total, working towards 120. You can even make yourself, there's the total for the content. 4 working towards 120. And this one, there's not a total, there's not a little sum there, so we can quickly auto sum and say I want it, well, it is already. I want the sum from there up to there. It will tell you we're on 4 out of 120 so far. So this is and I'm not going to save this because I want to send you a clean one. If you go, the 60 marks for grade 10, there's the test grid, that was the 11 test. Let us just see. Yes, here's the one for grade 10. Same story, same principles, but down it's out of 60 for the grade 10 test. So all these marks have been converted. The norm, remember the norm percentage stay the same there. That difficulty level norm percentage stay the same, but then the marks out of 60 is of course different from that out of 120 or 150. So then it's 24 out of 60, 15, 12, and 9 for your 4 different cognitive levels, and the same there for your difficulty levels. And the last one, we will also provide for those of you, and you have received this earlier, I know that, but if you're setting fully fledged papers of 150, there's a grid for that also, which gives you the cognitive levels marks for a test, then out of 150, or a paper 1 out of 150, and a paper 2 out of 150. So you will receive 3 of these cognitive grids or taxonomy grids in the next day. I'm going to send it tomorrow morning. You will receive the guidelines in the Free State. You will receive the guidelines as well as these 3 grids. I'm not going to save because I want to send you the clean one. Right. I nearly asked, are there any questions? But that won't happen now. Of course, you're welcome to phone afterwards or tomorrow if you don't understand some of these things. And please, it is not the cognitive, that grid is not given for nothing. When a paper is moderated, when a paper is pre-moderated, the grid and the memo comes with it. Before I go here, let me go here because the first part I will then open. Now, and let me try as good as possible to show you a few things, a few skills on the computer. And remember, computers differ. So hopefully this computer that I'm working on is the same that the one that I'm working on in my office or at home. So sometimes you struggle getting a route to a certain solution, but let's see how far we can go. It's just the basics to help you maybe just working a little bit faster on your papers. Now, type the paper and the marking guidelines into a template, or what some people call a macro. You can create an empty template. Now, this template is not the same thing as that taxonomy grid I just showed you. That's something different. That's a taxonomy grid or a cognitive grid or whatever you want to call it. This is a typing template. And I know there are some subjects that don't like this, that say, no, we are not typing inside these things. If you are comfortable without using this, I'm glad, but be careful, because things are jumping around in Word. They jump below, they jump on top, they get spaces in between them, and your aligning and things are not proper. But you decide. You can create an empty template of the paper in Word or the content grid, as I just showed you in Excel. Or you can use a given template, those that I'm going to send tomorrow, again of the paper, and I must add here the memo also. Create an empty template of the paper and marking guidelines. I'm just going to abbreviate it. And the same here, or you use the given grid template of the paper and the marking guidelines. Again in MS Word, and the content grid in Excel. Then, as I say, you type and draw, even draw your graphs as well as your diagrams inside these templates. And if you want to, because it's your paper, it's your school's paper, you can add a logo, as well as, of course, headers and footers. Headers and footers, we really like you to do that, because learners, they became used to our provincial and national set papers, and they have also always have headers and footers. I will show you what I'm talking about and how easy it is to add them. The logo is the school's logo. That's optional. You can add it or not. It's up to you. Now I'm going to show you the three templates that I'm referring to. The taxonomy grid we already spoke about. But let's go to the paper and then the marking guidelines, so-called template. And in red, I say it will be sent to schools. Right, so what I mean with a grid, not a grid, a template, or a macro is this. Let me just make it smaller so that you see this is a real paper. You can see this is a real paper. Look there. It's a real paper, but it's blank. It's blank for your convenience. The front page, you can do it as you wish. Name of school, logo, grade 11, life sciences test, November 2021, total time, as long as this information is there. The order and the way you do it doesn't really matter, but as long as this information is there, and it tells the learners this question paper consists of 13 pages. You can, while you're setting and while you're busy, normally you do this right at the end when you know exactly how long your paper is, and you find that here in the left-hand side bottom of your screen, your computer screen, which says we are now on page 1 of 13. Then you know the maximum number of pages is 13. Right. Just for interesting sake, some people want a border, this double-lined border around their first page, to put it on or off, and I'm gonna try to make it slow, but not too slow, because we are really limited for time. It's here on top. You see, there's a word that says design, and in design, on the right-hand side, you see page borders. There you can switch it on or off. You can choose for the whole document, only for this paper, for this side, whatever you want. If you cancel, nothing happens any further. So in design, you can switch on a page border. You can even change the type of border, a double line, or a thicker and a thinner line, or a thin line, as you wish. Then, let me go to the headers and the footers. If you're going to use these templates that we're going to send to you, the headers are already in there. And as I said, there will be 3 templates. One for the test, grade 11. One for the grade 10. No. There will only be one paper and one marking guideline. Sorry, so you have to adapt. Let's say you now you want to change this 11 test to grade 10 test. Sorry for people that knows this. Let us share with everybody. You just put your cursor inside that top part and you double click. Then you're inside the header. And you can write inside the header. Otherwise, when I double click outside, I cannot write there. I write here, but not there. So when I double click inside there, I can write inside the header. But when you change, the page number is a set equation. If I change there, the whole paper, the whole numbering system will change. But I can write there, I can write the date, whatever. What I normally do, and I must be careful now that I don't lose everything. Let's say you have nothing there on top. It's empty. To start a header and a footer from scratch, the footers will still be there, but the header is now gone. What I normally do, and please people, I know there's different ways and different routes working on a computer. Each person has his or own quick way in getting somewhere. I have my ways as well. I normally insert the page number. When you insert the page number, just press insert on top, you choose page number, you choose what type of page number, in the middle, in the center, and you choose on top in the center. And then because I say there's a tick on different first page, the first page won't start on page 1. It starts on page 2 and 3 and 4, etc. After that, I normally go back to my home on top and I put this page number on the left-hand side as not to center it. And then you can type that life sciences test story in there. And then with your space bar, you just move your 2 towards the center. If you want to know where the center is, double click outside. My cursor is centered. Just put a letter there, let's say small r. Then back to the header, and I move my 2 until it's right on top of the small r. And then I know that is center, and just remember now to delete the small r, because that can't be part of your paper. And going like that, now I can go also to the end of my document and put my November Free State, or if you can put your school's name there for that matter, UFS High School, no, not FS. We are working at the UFS. So it's UFS High School, and it's November 2021. And you see what will happen there, it will jump down. So all you do is you go a little bit back and you delete it upwards, so it comes back. November 2021. I'm going to spend a bit of time because people really sometimes struggle to put in the headers and the footers. If I go now to the bottom there, I can now say it's grade 10. And it's a test. Life Sciences grade 10 test. And when I double click outside, this information is on top of my whole document. Oh yeah, and the other thing that's important, remember I can do it at any page. This is not normal, it's always, you will see national papers also. It's in Arial, and the size is 10. Your headers and footers are in Arial 10. Now immediately you saw my things jump back. Now I must rearrange them and put them in the center again, like I did before. But it's Arial 10. The rest of my document, very important, is in Arial 12. If at your school you're using different fonts and sizes, we are not going to complain about that. The moderation will still pass, but to get your learners used to the provincial and national formats, start using these formats. Arial 10 for the headers, The footer is the same story. You double click inside the footer, you write copyright reserve, you write please turn over. If you have to translate, the Afrikaans schools, when you have to translate, the best way to translate is you do your English paper first. You copy the whole English paper to a new document, or you save it under a new name, and you start translating from the English document. But then here you have to go in and write kopiereg voorbehou, blaai asseblief om, ensovoorts. Right. So those are the headers and the footers. I hope I went too quickly. Let's see if my 2 is still in the middle. I don't think so. No, it is not. So I'm going to put my R there again and just bring my 2 back a little bit up to the R and then just delete the R. Instructions and information, it's given, so you can use it as is. These things you see in all our life sciences papers. And then the so-called macro that I'm referring to. The macro is this whole setup. But then inside the paper itself, you will see little dotted lines in the form of tables. It is nothing else but tables. But how do I switch it on and off? Because sometimes, let me quickly do it. Sometimes you find a paper like this. There's no dotted lines, but it looks perfectly correct. And now, how on earth do I get those thingies back? And you struggle and you suckle. Your cursor must be inside one of these tables. Let's say next to the A, because then you know you're inside the table. You go on top, when you normally work in the home, what are these things called? The ICT people will know. The drop-down menus, the titles of the drop-down menus, I think they call it. When you see home, you go right to layout. And then you see view grid lines. When you put it on, my grid lines are back. And keep it on, don't switch it off. Some people also work with these hojas on, these thingies. I do not. They confuse me quite a lot. If you prefer to work with them to show you where the spaces are, where the gaps are, where the what is, I switch them off. Okay. Now these macros, these templates, can you see my multiple choice, they all look the same. These spaces, 111 is in a little column. A, B, C, D is in a column. My answers is in a column. My distractors. My stem is in a column. And just remember when you type there, now I'm just going to do that, just put a space underneath to get a space between your stem and the ABCDs. And then you can type there. And when you enter, nothing strange happens. You type, and there's your distractors. And then you go to 112 and you do the same. Copy and paste works perfectly. You can copy distractors from another paper or somewhere else. You right click, you copy, and when you put it there and you paste, it pastes perfectly inside the next one. Just as an example of copy and paste. We've already put in for you things like questions so and so are based on the following diagram of, and then you have to include the diagram or the graph or the table or whatever, or the article, and then you ask a question, and there's the question and the answers again, A, B, C, D, inside your template. Scrolling down to the biological terms. Don't forget, let me just remember one thing. If your multiple choice are now only 6, you have to change here on top, it says number 111 to 1110, then you have to change that then of course to a 6. And then the example will be 117D, for instance, as an example. Remember that in the other sections as well. Biological terms, same story. There's the stem already. Afrikaans people, I will, if I can lay my hands quickly on Afrikaans templates, I will include the same thing as here in front of you in Afrikaans, so that these stems you have already in Afrikaans as well. Don't forget to justify those things. If it's unjustified, look what happens. Then they end like not even there at the back. So when you select, The minute you go more than one sentence, if there's more than one sentence below one another, you can select the whole lot, and then justify is there on top. Those little shortcuts, left align, center, right align, and then the last one is justify. It looks like a square block. When you press on that, everything jumps into square. Column 1, column 2, same story. You can type there, you can type there. Nothing, you lose nothing, nothing jumps to the left or to the right, unless you are typing inside there in the 131. Then of course, if I go there, I will destroy that. Or if I start moving these columns, look there, then this happens. So don't play with that if you're not familiar doing that. Right, and then the totals of each sub-question. Then 1.4, as I already explained, and just for the sake of neatness, or what can I call it, for the user friendliness maybe, normally we leave 1 or 2 spaces on top. When we start with the next page, 2 open spaces, or sometimes 1, it depends on your preference. There's 1.4 and 1.5, and then you go to section B. And at the end of section A, the total of section A is 40. So this was now for the grade 11 test. So if you want a grade 10 test, then you just change that 40 to 20. You replace that 40 with 20. Section B, there's questions for section B. Now, I like to put the diagrams or graphs or tables what I use inside the template. Then you know they won't jump around, you won't lose them. Even if they are not grouped or grouped, you know what I'm talking about. But many people, they put they delete these now, so that the background of the explanation of the question is just that one sentence there, and then they go here and insert their diagrams. It's okay, but then make sure your diagram is centered and all those other nitty-gritties. So I prefer putting a diagram or a table inside my template. So it is here, and then I move my questions up so that 211 starts there. And look at the distribution. 2.1 always totally right-hand side. 2.2, the main sub-questions. 2.3, etc. And then 2.1.1, 2.1.2, 2.1.3, etc. immediately aligned here in a column. And my marks are immediately aligned. You must now just, if you put in new questions, of course, adapt your marks according to your question. In the template, we already give you the format of going 2.1.1, which of the following, what, or give labels for, and then A, B, and C. There's small typing things that we have to discuss. We did it in the training in the past by saying, when you type here, when you start there, give, let's say, labels for, then there's a, what do you call that thing in English? I forgot. Never mind, you know what I mean. And then here you start again with capitals, with structure, say maar A, not bold, structure A, that is a capital again, but no full stop, because there's a, I won't get to that thing's name now, but never mind. Structure A, colon, A colon, yes. Colon and a semicolon. Semicolon, if I'm not mistaken, is that, a double punt. A double punt and a comma punt in Afrikaans. All right, so this is not bold. And then you can put in other questions there. So that if, like in multiple choice, you say the organ responsible for digestion is space, dot, 3 dots, and then your answer starts with a small letter. The structure responsible for digestion is A, or in multiple choice B or C, whatever, then that is the format in which you write it. Right. Just using letters from the keyboard there to explain something. I think that is more or less it. So question 3 and 4 and question 2 and 3, exactly the same. At the end of question 2, the whole of question 2. And remember your subtotals. Let me go there. 221, 222 and 223 is 2 marks, 1 mark and 6 marks respectively, totaling 9. And that is in bold. Right below the 6, it is the 9 is the total of 2, 1 and 6. 2 marks, 1 mark and 6 marks gives you 9. The same with the rest. And when you come at the bottom of question 2, the whole of question 2 is 40 marks. You can put it in those type of brackets. You can even remove those type of brackets. That's not the end of the world. And that means some people write here total question 2, 40. No problem. No problem. Refer to the national papers also if you want to move towards the national papers or the provincial papers. Same with question 3. And then right at the end of the paper, you will have now total for section B is 80. And the grand total for this paper is 120. Now there's an interesting thing. How do we remove this? Please turn over on the last page, because learners cannot turn the page again and again and again. They have to stop turning the page. The easiest way, there are different ways. The easiest way, remember now this is in the footer. So if I just delete it here, look what happens. If I delete it here, it's gone on all the other pages because it's a footer. So let me undo that so that it comes back. Please turn over. I just want to remove it on the last page. Easiest way is to insert a frame, like a shape, not a frame, a shape like a square. You insert a square over that whole thing, but then you have to change the format of the square. So you go right back on top where the shape fill is. You say, I don't want it to be, no, I want it to be filled with white. And then the outline is nothing. And then the page turned over on the last page is gone, but it's still on the second last and the other pages. That's how you do that. If you forget it, that is not the end of the world. I'm finishing off by going quickly to how to draw certain things in a life sciences paper. Now, in the things that you're going to receive tomorrow, in the guidelines for setting papers, there is a part at the end, how to draw tables, graphs, and diagrams. And then there's descriptions, there's explanations. Look at it, use it, go through it, and the only thing you will manage this or getting masters of these things is to play with it, is to use it in your papers, in your documents that you do. So play with it. Take another document, play with it, delete it, undo it. So there's also, when it comes to graphs, there's this nice big diagrams showing how to do this things. I'm quickly going to do all 3 of these things for you, and then but you must go and practice it. Then there's a last part on moderation. What is moderation? Who moderates? It's important. And then we're providing you with a checklist. So your departmental head or your colleague in a neighboring school use this tool to check your paper beforehand and give it back to you with, yes, it is there, no, it's not there, and recommendations. Very simple, short tool for pre and post, for pre-moderation. Post-moderation, when you moderate scripts of learners, there is another tool that you have in your subject assessment guidelines, of course. Right, so this information is in the document that we will send to you. So let me start by showing you, there's a table somewhere here. I saw it, okay, that is also a table. Where's that table? Here's a table. So to insert a table, you see, I'm outside the template. Doesn't really matter, it's still there, but if you do it, this one is already been shifted or moved so that it's in the center. So let me do that same table in the template. I'm going to keep that original one at the bottom, and I'm going to try and draw that same table now here. Right, for those of you who struggle with tables, I know there's various ways in drawing tables, but the easiest way is insert. There's a little icon that shows a table. You click inside it to choose how many rows, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 rows and 2 columns. So 1, 2, 3, 4, 5, 6, 7, 8. Let's just use 8 rows and 2 columns, and if I click there, there's my table. It looks more or less the same, although it's not the same thickness. That can be changed. Mostly our lines in life sciences is of 1.5 thickness. So what I do is I select the table, I choose right click. I know there's different ways. I go to table properties, I go to borders, and inside borders I can just choose another border like 1.5, 1.5. Here you can take away some of the borders or add, but I want that original table 1.5 thick, and when I press the 2 OKs, it is like that. So for starters, do a table like that. Don't bother with thicker outsides and thinner insides. Yeah, you can play with it, but if you are pressed for time, just go for 1.5 thickness and you will be okay. So if I delete that, I have just 2 rows less, but at least you get the idea of drawing a table. To insert rows in a table, even that one or this one, I know there's easier ways, but I normally choose right click, I insert, and then I choose below or above. It's below, the row jumps in below, or above, it jumps in above. So in my table, my cursor must be inside my table and where I want the row. If I want a column, the column will also tell you left or right. So right click in a particular column, insert columns to the left, will add another column, and in the same way you can delete a column. Right click, there's delete cells. I want to delete an entire column. Yes, then that column is gone. So very easy to draw tables. I want to delete, I only want 3 rows in my table, so I select those I want to delete. I delete, choose them, and I delete them all. Now I only have 3 rows and 2 columns. But as I say, there's numerous different ways in doing this. Simpler and quicker ways, I know. Maybe I took a longer route now. Right, that's tables. Let's now go to graphs, which is a bit more difficult. Let's go to question 1. I want a graph. The graph below shows what? Something that you're going to type there. Now, what is this graph going to show? The easiest way for me to insert a graph is you go to insert on top of your Word document. You choose chart, and then you will have options. You will have columnar bar graphs, line graphs, pie graphs, whatever. There's a lot of. The easier one, the columnar, and then there's a different forms of columnar bar graphs. The simplest one is the one right at the beginning there, the first one. Here it is, and then if you say OK, there will be an example of a graph in your document. You can move this away if it's in your way, but that is the information of your graph. So again, I say, yes, you can select a certain table and then just insert that graph, But there you have a general columnar bar graph. If I only have one variable that change, you can go inside that data, and this data is in Excel. Can you see it? It's green. It's in an Excel document. So immediately when I want to reduce my variables, I just drag there from right to left, and when I leave it, now these are the graphs that we normally see, the ones with the one column or the two columns. If you have a double barrel columnar bar graph, you can go that route. But let's make it simple for this explanation and go to only one bar. These two, because I've only selected this two, it will stay like that. Otherwise, you can delete. If you're afraid those two are going to make trouble for you, you delete those two. They're gone. And now, category 1, category 2, and 3. Look there on my explanation. There's now also category. Let's see if I can enlarge a little bit. Here it is. Category is at the bottom of my columns. So I want, in my particular graph, months of the year. January, February, March, and April. Then I physically type January, and you can see it's happening. When I go to the next one, it's happening on my graph. January, that one is now February. Then the next one is March. And the last one is April. And when I'm done, they're already in my graph. When I close that, I'm not going to do it now. You can come back by saying editing your things. But when I close that, my graph is in my paper. And then let's say, forget about the titles, the X and Y axis titles. Let's say this is rainfall. In January, there was only 5 millimeters of rain. Look what the graph is doing. Then in February, there was 20 millimeters of rain. And then in March, there was 15. And in April, there was again only 9 millimeters of rain. Now, if I close that again, let's do it now. My graph is in my paper. Not like I want it finally, but at least months and rainfall figures are there now. Now I can go and edit my graph. So if I click anywhere in this graph, now nowadays the Word formats, the Office formats have the editing right here at the right hand side. So if you press plus, it shows you I can edit axes or titles or whatever. In the older versions, it's on top there. You format your graph and then you work there from the top. Okay, at least I want a chart title. I want axis titles. And I want data labels. I'm going to explain now. The data labels are these on top there, the 5 and the 20 and the 15. Sometimes in a paper you don't want to show learners that they must read from the graph, then you don't put it in. Otherwise you can put it in. In the series 1 there, that's my title of my graph. So here you can physically type. It's a graph of rainfall in different months of the year. I'm not going to type because it's going to waste time. Rainfall figures for different months of the year. 2 variables in my title. Okay, series 1 is my legend. I can just delete it, then it's gone. Then here I want to write months of the year, and there on the y-axis I want rainfall. Rainfall in millimeters. My label is in brackets. But what you also see is these things are in gray, so I can just click there. I can go to home. I can choose, as I normally do in a Word document, and I choose black, and all these things becomes black that everybody can read it. You see what I do? Even my figures. And I can change the font. If I want that also to be Arial, and normally it is Arial 12, like the rest of my document, I change it like that. Change the y-axis label to Arial 12. And change the months, the word months, to Arial 12. And change the February and January to black at least, and then Arial 12. If you have not enough space, you can use other fonts, but this is the standard fonts used in graphs. If I want to, normally in a graph, there's a nice visible line there on the x-axis and on the y-axis. In some way, I must go in like I did just there. You struggle sometimes to get in. If I go out again, and I go in, the second time lucky, it forms a square, a block around the months, and then I say I want to work on my axes, so I choose axis. No. I choose the different options. Okay, I've removed now that. I must go there to different options, more options, and here comes a lot of new options, and there's the line colors. I choose line. It is currently gray, that's why it's not visible. I choose black, and I choose 1.5 thickness. And you see when I go out of all of this, let me somewhere do the y-axis as well while I'm here. I can do the y-axis, black, and 1.5 thick. And now when I go out, 1.5 thick, when I go out of all of these, click on the outside, there is that horizontal and vertical lines. To get this thing out, there's also an option. You go to options and you choose, I don't want that tick mark there or that little line there. You can switch it on or off as you wish. To make this bigger, when you click there, it chooses all those thingies, and you can choose another font there on top. Or you can just make it, let's say, bold and black so that it's more visible. And then to change the color of your graph, when you click there, and you can, when you right click, it gives you format data series, and you go to color, but now it's the fill color and not the border color. I want solid fill, and I want, let's say the first one. We normally make it black or a dark gray. If you make it dark gray, it looks like that, but the paper normally is black, and learners don't have colored the paper, so you can't make it yellow and green and white, but for your own purpose you can. And there, more or less, I have a graph. The basic things that you need to draw a graph is now in there. The grid lines, you can switch on and off these grid lines. It is with axes, it is with axis titles. Where's the grid lines? Let me choose the right click, format plot area. Somewhere here you will have grid lines when you choose the graph itself. Well, let me just do this. Let me go there. Choose data series. I think there I will be helped. You see here the gap width. If you close that gap width, by the way, it becomes a histogram. Otherwise, you just go to graph and to change to select or remove the grid lines. Here's grid lines. I can switch it on there, on and off as well. So when I switch it off, there's no grid lines. If I choose grid lines, I can use even smaller grid lines, you see. And then it makes it smaller, vertical and horizontal like that. Or you can just switch it off and then they are gone. So grid lines are in this thing on the right-hand side at the plus sign there. Right, I think I'm going to step off from graphs. That is the basic way to draw a graph. Now, when you want to change the type of graph, you can do it now or rather start from scratch. Let's see if I go there and I change the type of graph to a line graph, what it does. Then it's the same information, but in the form of a line graph. But to draw a line graph, rather start from scratch with the new information that you need. In the same way, of course, you can change the chart to a pie chart. And then you can play with your colors and your segments and so forth. Right, I want to go back to my bar graph. That is the one that looks the best of them all. And the last thing I want to quickly explain to you, we have a few minutes left, and that is diagrams. That's another training on its own, but you're fortunate if you get a diagram like this. When you click in there, you see these things are all grouped, but they consist of different separate diagrams. There's one diagram of that flower, there's another diagram of that flower, there's a diagram, or there's a shape rather, for that label line, there's a shape or a text box for the A, there's a text box for the B. Now, you're fortunate if you get a graph like a diagram like this, because then you can go into the diagram and you can change. Not the drawing itself, but the labels. I mean, I can remove the label B totally by just doing that. Then I only have label A. And I can play with, I can remove the B. If I want to put in a caterpillar, I can remove the B and put in a caterpillar. Or put in new labels. How do I put in new labels? I have now label A, but now I insert a shape. Remember, it's a line without an arrow. And I just draw that line. Now it's in blue, so there's a little bit of work to do. So I can change the format object by just changing it to the line, to a solid line, black, 1.5, not there, width, 1.5, I see what it does. It's not the label that I'm working in, it's the frame on the outside that I'm working on. So be careful, just be careful for that. You only choose the label line, and then you do the same story. Line, solid line, black. Hopefully it will now just only the line becomes 1.5 thick, and there is a label line. And then you insert a text box, as I did that. So I can either insert or just copy that one and paste it. So if I draw that pasted label now here, that can now becomes label B. All right. When my diagram is smaller, then it will also look more in the way that we draw diagrams, because it's so largely enlarged that you see it as very thick lines and big letters. So you're fortunate when you get it like this. You can play and add and delete inside this diagram. And when you're done, you can just group all these things so that they don't jump around. If you choose the text box there, hopefully it will do it now, and you press Control, and remember when that thingy becomes a plus there, okay, the diagram has changed to another format, so that, okay, these things are grouped, but now I just want to group this new additions of mine. So I choose those by keeping in Control and click. So I choose B, I choose that line, and I choose, let's say now, the diagram on the outside. Rather start with that, and then hopefully go to B and to that line. And when I right click and choose group, they are all fixed. If I delete this group diagram now, everything will delete. If I can just get my cursor to pick it up. If I delete, everything is deleted because everything is grouped. So that's another function that you really have to play with, is grouping your diagrams. There are other ways, for sure. There's other ways than the one that I just showed you. I quickly want to show you. Now I got the same diagram, and I purposely just snip it. I will show you how to snip. To get everything, now I can't work inside this diagram. It's a whole picture diagram. If I press delete, everything is gone. And I can't go inside the A or the label line. It's impossible, because it's a total picture that I maybe got from a textbook, I scanned it, or I got it from my notes or wherever. Now I want to change things in this. I need to go to Paintbrush in MS Word to work on this diagram and change things, and then bring it back, and then add my labels. So I need, just to show you quickly, I only want the B. I only want to take the B from this diagram. If you click left bottom, and you type in there snipping tool. Many people have snipping tool here on the bottom menu. And snipping tool asks me a new snip. Then everything becomes faded, but when I choose the B, I only cut out the B from this diagram. And when I put it in any new or other diagram, let's open a new Word document, and I right click and I paste, there's my B. I took it from the previous diagram. Yes, I know there's other lines and stuff. I can remove that in Paintbrush. But it's just to show you how to get a part of my diagram. Now, doing it like this, any past paper, let's say in grade 10, you want to draw a diagram of xylem. You take in any past paper only the xylem, you snip it, you bring it into your new paper, you paste it, you have your xylem, and then you can work on it. But I want this whole diagram to work on. So if I right click and I copy this whole thing, and I don't see Paintbrush here at the bottom, so I also go left hand bottom, and I just type in Paint. It will bring me Paint, the application. I open Paintbrush, and everybody, I think, that's watching this afternoon knows how Paintbrush is working. You can either paste or right click paste. There's my diagram that I work in my paper. I can enlarge it a little bit as to see it bigger, to particularly work on things there. Now I can, for instance, I can choose select. I can, for starters, delete the frame that is here. I don't want the frame. So I delete the total frame that is too thick and doesn't look neat. I want to remove the B now, totally. I want to remove the B, I want to remove those label lines. Up to there, I can use the select delete function. But the smaller things, I must go to my eraser, and now I can erase smaller things. And because my diagram is so enlarged, when I make it smaller, you won't even see that the B and that line was erased. So in the same way, I can maybe delete because what happens if you take all your labels from Paint, it won't be in the same format. In other words, it won't be Arial 12 in your document. If you just take a picture, look what I've done. I've removed now all, it's too small. I can't work there when it's so small. I want to remove that label line. Unfortunately, you can make the eraser smaller. You choose those lines on top there, you choose the small eraser, and now you can erase that small thing is there, and even that label line there. So more or less, label lines deleted. Now I only want to take one flower. Let's say I only want to take one flower back to my paper. So I choose select, I choose my flower, and now I must press copy, because I want to copy it to my paper. Yes, no, this is not my paper. There's my paper. I don't like this diagram anymore, so I delete the whole thing. I want to work inside where I was previously, and when I paste here, what will happen? Only that one flower will appear here. And now you see it's a picture. Normally, when you want to work and put labels in, change the format of that picture to something else. Like here, you choose the wrapping of the picture to either square or tight. And then you will see I can even now move around easier, and I can put labels inside and even change, and afterwards group. Otherwise, I won't be able to group. So last thing is now how to put in, let's say, a label line. You can go to Insert Shapes. You choose a line, and I want that corolla or whatever to be a label. And now just in the same way, I go to fill, make it black, make it 1.5, and then I have a 1.5 label line there on my new diagram. To insert a new label, you go to Insert Text Box. You can either draw the text box or just choose a text box. This must be labeled Q, type in the Q inside that little block and move it there, and there's my label Q. It still has a frame around it. I don't like the frame, so I choose shape outline there on top, and I don't want any outline, and there's my Q label. Goes to the label line. I can even move this one closer or further away to that line. Going in front of behind, that's another story and another day's training. But now, remember, now I have my flower, I have one label line, and I have a label, Q. I want them all. If I only delete now that, what will happen? Only the flower will delete, not those other two. So undo, there they are back. I choose my flower. Control, I click there on the flower. You see the dotted figures on the outside. I press Control on my keyboard and I choose the label line. Now, why don't that one wants to, let me go first to the label line, then to my flower. Okay, there's something wrong here. It's the thing of is it in front or is it at the back? How is this one wrapped? So I can also choose maybe the wrap option with text or whatever, and hopefully it will choose it now. Not yet. So there's some things to play here to group it. The idea is when you press Control, it chooses all your thingies, even the label line there, and you can group it. Let me just quickly go there, do that also. I don't know if this will work. Make that also square. I know there's other ways, and I know there's a very quick way. I make that also square, and let's see if I can maybe group them now. No, not yet. So it's that front and behind, it's the arrangement, the order in which the diagrams are here on my diagram. That is not now 100% correct. I can also select all objects. Selection pane, I can choose here to select the text box and the connector. You see there, they choose it. So that I can afterwards then just choose group. So this the thing that I want to explain here, and it will take me a while just to get them grouped. As I said, sometimes it differs on different computers. There's a step that I'm just now missing as to group these diagrams. But then you can group it. And then at the end, you normally see that our diagrams have frames around them. To insert a frame, you just choose your diagrams, your shapes. You choose the square. You put your frame around everything. But now it's not visible, so you must make it visible as to say no fill. And the outline, remember, it's black, and the weighting is 1.5 again. Now I have a frame around it, as we see it in papers, but it still needs to be grouped with the rest. So at least I should be able to group my frame with my flower. I think my problem is my flower. My flower needs to be in another format. I'm not going to struggle with that. You know me, I want to solve it now. And when I drive from here, I will know exactly how to do it. Normally that's how it works. So my wrapping of my flower is toch square. Why don't it want to group the rest of the thingies in any case? So there's the diagram. If it's inside the template, it won't also change. I mean, I can enter there on top. Look, the whole diagram is going with. So that is also a way, maybe it should happen now like this afternoon, is that I can't group. I'm struggling to group. And now when I work outside my template, I can still play around and enter spaces and delete spaces, and my diagram stays exactly the same. But when it's here outside, sometimes when you enter or when you delete, then the Q or the line or the frame moves away from your diagram. So it is a safe way of keeping it inside. Right, we are about 13 minutes past our time limit. I think at this stage, viewers, teachers, that you will be able to set a well-set paper for the end of the year, grade 10 and 11, using these principles. And from my side, yes, let's go back to the camera. From my side, I hope you've learned something this afternoon. I hope that you can use some of these things. So please make sure that your end-of-year papers are of a good standard, that you applied all these principles that we spoke about this afternoon, and that you will be able to apply. Feel free, after you have received these documents from us, feel free to contact any of your subject advisor or even myself, and ask more questions on these topics. And we will be more than willing to help in whatever you need. So from my side, go well and good luck towards the end of the year. Bye-bye.